right, so we're going to change gears a little bit and talk about work we've done looking at low-input ChipSeek. And so ChipSeek is a technology for mapping protein-DNA interactions, such as the post-translational modifications of histones. And it can be useful for studying both gene regulation and finding regulatory elements, such as enhancers. And so different elements in the genome, such as promoters and enhancers, are marked by different combinations of post-translational histone modifications. And the unique combination of these marks can tell you whether a gene is typically on or off or in an in-between state. So the ChipSeek workflow is simply you cross-link your protein-DNA interactions using formaldehyde, you sonicate your genome to fragment it into fragments, and then you use an antibody to immunoprecipitate your protein and its bound DNA. Then you purify the DNA and deep sequence it to identify where your protein is binding DNA. So traditionally, this assay works with inputs in the millions to 10 million cell range. However, we were interested in improving this down to around 10,000 cells, which will allow us to use samples we get from clinical populations. So again, some of the limitations for pushing the ChipSeek assay down to low numbers are, again, traditionally it requires high cell numbers, and that the efficiency of enrichment for where your proteins are binding, it can vary from 1 to 50%, and most of that is dependent on the quality of your antibody that you're immunoprecipitating with. And then in the library preparation, it can be inefficient due to the really low amounts of material you often get post-IP that are in the picogram range. There's a broad bimodal size distribution because of the sonication, and then a size selection to reduce the distribution often results in high sample loss. So all of these are challenges to pushing the ChipSeek assay down. So we use the kappa hyperprep kit, as Adriana mentioned, and it simply starts with DNA, and it uses a single tube to do the end repair, A-tail, and adapter ligation reaction, followed by one spray cleanup, a PCR amplification, and then an optional size selection, and you're ready to sequence. So the whole protocol can be completed in one workday. The hyperprep kit requires currently for ChipSeek a recommended 1 nanogram or more. And so what we wanted to do is, if we're going to push this assay down to 10,000 cells, what is our input range going to be? And so we started performing some chips in the lab, and we're going to use the H3K4 trimethylation. It's a histone mark that's at all transcription start sites that are active. It has extremely high quality reagents and a very defined cellular distribution at transcription start sites, which will allow us some nice QC metrics down the line. And so when we do the ChipSeek for this, or chip for this mark in Raji cells, we get a very nice enrichment at positive control loci and nothing at negative control loci. And using 10 to the 7 cells, we estimate that we get about 30 nanograms of DNA pulled down. And this is from about 10 different IPs. And so extrapolating down to 10 to the 4, we think we're going to be in the 30 picogram range. And that assumes that our enrichment efficiency stays the same as we go down and push the assay further. So if we look now at our sequencing prep protocol, we decided to take 1 nanogram, which is the minimum requirement the kit recommends, and titrate down to 100 picograms and 10 picograms. And the way we're going to optimize the assay is focusing on the adapter ligation step. And so we devised this matrix where we would increase the amount of adapters to try and see if we can drive conversion efficiency or the amount of adapter ligated library we have at the 1 nanogram, the 100 picogram, and the 10 picogram at these different concentrations of adapters. So we took our 10 to the 7 cell chip and diluted it to these volumes so that we were working with the exact same IP, and that would allow us to compare our results that should look the same. And our success metrics were basically what maximizes our conversion, the amount of library we're getting, and with minimizing the amount of adapters that are carried through to the sequencing, which can negatively affect our sequencing run. And so some of the QC metrics that we do, as Ben alluded to, is that post-ligation, we use the kappa library quant kit to determine exactly how much adapter ligated material we have. And this allows us to empirically determine the number of PCR cycles, so you minimize bias due to PCR. And importantly, it allows us to perform a melt curve analysis, which is diagnostic for how much adapter dimers you have post-ligation. So here is a PCR post-ligation data for a high complexity library with no adapters. You can see that the melt curve peak is below 80 degrees, whereas if you just ligate adapters together and look at where it melts, it's above 80. So doing a melt curve, we can start to look at the complexity of our post-ligation data. So quanting how much converted library we have showed that increasing the amount of adapters at 10 picograms, 100, and 1 nanogram increased the amount of converted material we had. And you can see significant improvements at the high adapter amounts for 10 picograms and 100 picograms. So how much of this was adapter dimers and how much of this was actually converted library? So at the 300 nanomole for 1 nanogram, you can see a double peaking here, where the left here represents adapter dimers, and this is our converted library. And as we increase adapters, we're going almost to completely adapter dimers. And this is true for our 100 picogram and our 10 picogram. So we know we have a lot of adapter dimers that are being carried through the protocol. And so we still went through and calculated how many PCR cycles we would need to amplify each amount for, knowing that we're probably estimating this number. So again, after amplification, you perform a spray cleanup. We then performed a size selection, and we pull samples post-cleanup here and post-size selection and perform both the kappa library quant and a bioanalysis to visualize adapter dimer carry-through so that we knew if the size selection was removing it or not. We could estimate sample loss and identify other PCR artifacts. So these are bioanalyzer traces for the 1 nanogram input. In red is the post-PCR bioanalyzer, and in green is the post-size selection. So you can see how in green the post-size selection really narrows your distribution nicely. And here in the highest adapter amount, this little red peak right here is adapter dimers that were present post-PCR. And you can see that they're completely removed after the size selection. We can again see this in our melt curve, where in gray is the post-PCR, and you can see a little adapter dimers here, but everything is shifted to the left of 80, indicating there's no more adapter dimers after the size selection. At 100 picograms, you can see adapter dimers increase as we increase the amount of adapters, but in every case, it's removed by the size selection. And in every case, we start to nicely select for a very single size distribution. At 10 picograms, you see at the highest adapter amount an enormous amount of adapters that are present post-PCR. And again, it's completely removed by size selection, but you also start to see this double peaking here and in here, which is can be over amplification problem. And so these are samples that were probably PCR'd too much. And so this is show you how you can sort of diagnose PCR amplification, over amplification at the bioanalyzer before you sequence. And so here is the final yields for each sample. So the 1 nanogram, we increased our final yield post-size selection up to using 1.5 micromolar adapters. Again, at 100 picograms, the 600 nanomole adapters gave us a big increase in yield. And at the 10 picograms, we maxed out our yield around 150 nanomoles of adapters. So we chose the highest performers, which were these three. We technically replicated those libraries to see if what the replication was for this, and we sequenced on the Illumina platform all 6 libraries. We sequenced them all to similar coverage, and they all had very high mappability rates. If we look at our clonality and the duplication rates in each library, that the 1 nanogram samples are extremely diverse and complex libraries. But as you go down the input amounts, you start to lose your complexity in your libraries, such that in the 10 picogram samples, only 7 to 2% of your total reads are actually unique. They still, despite this problem, similar numbers of peaks are called in every case. And if we start to compare technical replicates, you see a very nice high correlation with the 1 nanogram and the 100 picogram replicates. But you start to see a little bit more noise in the 10 picograms, but these are still highly correlative. If we just look at the overlap in peaks, between the 1 nanogram and the 100 picogram, 93% of them overlapped. And between the 1 nanogram and the 10 picogram, 61% overlap, suggesting as you go down, you start to miss and have a lot of false negative rates of things that you would pick up if you used more samples. If we start to look at correlation plots between both the 1 nanogram on the bottom and either the 100 picogram or the 10 picogram sample, we still see a very nice correlation of signal intensity or read depth at every peak. However, you might have noticed that there's this arm that starts to come out, especially in the low input samples. And it's peaks that are sequenced that have a lot more reads in the 10 picogram than in the 1 nanogram. And when we start to look at what these are repetitive elements. So when you start to get really low amounts of input material, we're starting to sequence some junk DNA in there. They have a lot different GC content than the rest of the library, and they are not conserved. So you're starting to amplify some junk, although it's a very small amount of your library. So again, we wanted to apply this to a low input ChipSeek. And that's because we get samples from the clinic that we would like to perform ChipSeek on that are around 10,000 to 100,000 cells. And so we improved the chip assay in our hands that I'm not going to talk about, using both reduced volumes and magnetic beads, cleanups to reduce sample loss. And we performed chip in our cell line using K4 trimethylation on both 10,000 and 100,000 cells. You can see we maintain our high enrichment at positive control versus negative control sites. We sequenced these libraries, and you can see our 10,000 cell library still has 40% unique reads in there, with our 100,000 cell around 70%. The correlation at promoter regions for libraries prepared from 10 million cells or 100,000 or 10,000, you can see there's a very high correlation of signal intensities. And if you look at a genome browser screenshot, you can see that the peaks are almost identical whether you're comparing 10 million or 10,000 or compared to OnCode data, and that the peaks occur at transcription start sites for genes. And you can zoom in to one locus to see even clearer how the same peaks and the same transcription start sites are called.